Okay, my name is Kanta Suemitsu from Hokkaido University, and I first of all want to thank the organizers for giving me an opportunity to talk here. And so my topic is about the evolution of the gravitropism in land plants. And as you can see in the title, I use the moss Physcomitrium patents to analyze the function mechanism of the gravitropism. And so I think I don't need to explain this slide much, but well, just here, I want to say that in angiosperms, the amyloplast sedimentation is, well, what senses gravity, and the signal is transmitted by the lazy proteins. And as a result, it results in the differential organ growth. And that's the system in angiosperms. But then we can see a gravity response in all land plants, including bryophytes. So we can imagine that this system of the gravity sensing evolved after the terrestrialization because the gravity condition is really different from the water, inside water and outside water. But then because we don't know how the gravity is sensed in other organisms than angiosperms, we don't know how it evolved. So we decided to study the mechanism of gravitropism in the moss Physcomitrium patents. And so since I'm the first one to talk about this plant, I'm going to explain a little about the life cycle. And so unlike the angiosperms, first of all, it's different from the germination because they first germinate from spores and they produce a filamentous tissue. It's a single cell row of tissue, which is called protonema, and they eventually produce a 3D structure called gametophores. And the sporophyte stage is really short. The protonema and gametophore is both gametophyte stage. So the sporophyte stage is short. It's just to produce spores. And within this life cycle, the protonema and the gametophore are known to exhibit gravitropism. Well, they exhibit negative gravitropism, and they occur only in dark. So for this plant, light is much more important, probably. And so in this talk, I'm going to explain about what we found in the protonema, of the gravitropism of the protonema. So we will continuously from Professor Morita. Well, we focused on the function of the lazy and Physcomitrium patents. Well, Physcomitrium has 5 lazy genes, and the protein sequence similarity is really low compared to the Arabidosis. There's only about 20% similarity between the Arabidosis and Physcomitrium. But certain domains, well, probably most importantly, including the domain at the C-terminus, is conserved even in the Physcomitrium patents. So we thought that these lazy genes in Physcomitrium would probably also be involved in gravitropism. So we knocked out all 5 of them and produced a quintuple knockout mutant by CRISPR-Cas9. And we analyzed the phenotype. And so, as I said, we can only see the gravitropic response in Physcomitrium under dark. We put the plates in dark and place it vertically and waited for 2 weeks. And when we first opened it up, we were a little disappointed by the result because even in the lazy quintuple knockout mutant, you can see that this filamentous tissues, the protonema, are still going upwards. But then when we had a closer look, we could see that some of the filaments were, not straight upwards. And when we did the quantification, there was a significant difference. So, but here it's not clear. So we wanted to have a more clear result. And we decided to change the angle and track the change in the tip angle by rotating it by 90 degrees. And the result was much more clearer because, well, after 48 hours, the tip angle of the wild type was free oriented, like 70 to 80 degrees. But in the lazy knockout mutant, it was around 50 to 60 degrees. So there was a significant difference. And well, so by this result, we concluded that the lazy and Physcomitrium are indeed involved in the gravitropism somehow. And well, you might think that even in the mutant that knock out all lazy genes, we can still see a gravitropism here. And so you might think that there is a mechanism other than lazy that is involved in the gravitropism. And well, actually, we talked with Professor Haodong Chen in this workshop, and well, he said that he had a different phenotypes. So well, now we're thinking that this might be a weak allele here that shows weaker phenotypes in the gravitropism. But well, anyways, the Physcomitrium, the lazy and Physcomitrium was indeed involved in the gravitropism. So we wanted to address how. And so we knocked in a fluorescent protein at the C-terminus in one of the lazies, and we observed the localization. And at first we were hoping that we could see a signal at the plasma membrane and also at the chloroplast, because well, the Physcomitrium doesn't have an amyloblast, but then they do have plastids, they do have chloroplasts. So we thought that it might localize there, but we could see a signal at the plasma membrane, but we couldn't see any at the chloroplasts. And well, this is the basal cell of the protonema, and well, but the gravitropism in protonema, well, unlike the gravitropism in angiosperms, well, it's a single cell row. So there's no way the differential growth is involved in the gravitropism. It's caused by tip growth. So we looked at the tip cell, but then when we grew the cells, well, the moss under white light, well, we couldn't see any signal at the tip cell. We could see at the basal cells, but not at the tip cell. And then, well, this wasn't the first time we had a result that we didn't expect. So we decided to change the condition, and we remembered that the Physcomitrium only exhibits gravitropism in dark. So we placed it in dark and observed the signal. And well, when we placed it in dark, this is 3 weeks, and we could see a clear signal at the plasma membrane. But well, there wasn't any really polarization here. And this figure at the right is just to show that, well, even in this condition, we couldn't see a localization at the chloroplasts. And so from this result, we were thinking that the lazy abundance at the protonema tip cell may be regulated by light. Well, we don't know if it's the transcription regulation or after transcription, but yeah, we're thinking that the lazy is somehow downregulated by light. So as a summary, so from our results of the phenotype analysis, lazy seems to be involved in the gravity response of the Physcomitrium patents, but we still don't know how the mechanism, how they sense gravity and how they, we don't know how they sense gravity. And also, light seems to induce the downregulation of the Physcomitrium patents. And this may be related to why the Physcomitrium patents exhibit gravitropism only in dark. And lastly, unlike the Arabidopsis lazy, we couldn't observe the signal of the lazy at the plastid or at the chloroplast. So, well, this is just guess. We don't have any evidence yet, but we're thinking that maybe the ancestral function of lazy and gravitropism was independent from the plastid sediment, amyloplast sedimentation. And maybe it was short, but that's all I have today. And thank you for listening.